Glycolysis is a metabolic process that breaks down sugars to generate energy. This process occurs in two main phases, an energy-requiring phase and an energy-releasing phase. To begin with, the glucose first enters a cell's cytoplasm. Next, through a series of enzymatic reactions that require energy in the form of ATP, glucose is modified into two molecules of 3-carbon glyceraldehyde 3-phosphate. In the second phase, through multiple enzymatic steps, the two molecules of glyceraldehyde 3-phosphate are converted into two molecules of pyruvate, 2-NADH, and 4 ATP molecules. Both these pyruvate molecules are then transported to the mitochondria for entry into the citric acid cycle. The energy-requiring phase is the first phase of glycolysis. As glucose enters the cell, the enzyme hexokinase transfers a phosphate group from a molecule of ATP to the 6-carbon sugar that produce glucose 6-phosphate, which becomes trapped inside the cell due to its negative charge. Next, the enzyme phosphoglucose isomerase catalyzes the conversion of the phosphoglucose into one of its isomers, fructose 6-phosphate. The phosphofructose can now be phosphorylated by a rate-limiting enzyme, phosphofructokinase, to produce fructose 1,6-bisphosphate. Finally, with two phosphate groups attached, the sugar molecule is cleaved by aldolase into two 3-carbon isomers, glyceraldehyde 3-phosphate, or G3P, and dihydroxyacetone phosphate, DHAP. Another enzyme, triosphosphate isomerase, converts the DHAP into G3P to yield two molecules. This way, during the energy investment phase, a net total of 2 ATPs are used to split one initial glucose molecule into two smaller sugars. The second phase of glycolysis begins with the oxidation of two molecules of glyceraldehyde 3-phosphate. It is catalyzed by the enzyme glyceraldehyde phosphate dehydrogenase, which transfers a phosphate group to the sugar, forming 1,3-bisphosphoglycerate. 2 electrons and a proton released during the reaction are picked up by NAD+, forming NADH. Phosphoglycerate kinase then transfers a phosphate group from each 1,3-bisphosphoglycerate to ADP, creating 2 molecules of ATP and 3-phosphoglycerate. Next, the enzyme phosphoglycerate mutase converts 3-phosphoglycerate into its isomer 2-phosphoglycerate. Further, the enzyme enolase removes a water molecule from each 2-phosphoglycerate to form 2 molecules of phosphoenol pyruvate, or PEP. With the help of pyruvate kinase, the phosphate groups are removed from PEP and taken up by ADP, generating 2 more molecules of ATP, along with the end product pyruvate. In this way, at the end of the energy-releasing phase of glycolysis, 4 ATPs, 2 NADHs, and 2 pyruvate molecules are produced. So, in summary, glycolysis can be divided into 2 phases, the energy-requiring phase and the energy-releasing phase. In the first phase, 2 ATP molecules are used to split the glucose into 2 molecules of glyceraldehyde 3-phosphate. In the second phase, the intermediate sugar is further catabolized to produce 4 ATP, 2 NADH, and 2 pyruvate molecules. Because 2 ATPs are consumed in the first phase, the net yield of glycolysis is 2 ATPs, 2 pyruvates, and 2 NADHs. But glycolysis extracts less than a quarter of the energy stored in the glucose molecule. So, in the presence of oxygen, pyruvate can enter the mitochondria for further oxidation. Once inside, pyruvate is converted to acetyl coenzyme A, which can then enter the citric acid cycle, producing more NADH molecules. All the NADHs then donate their electrons to the electron transport chain, producing ATP and completing the oxidation of glucose.